All right, so today's experiment, you guys, is that we're looking for the activity of an enzyme called catalase. And like I just told you, catalase breaks down hydrogen peroxide into water and what? Oxygen gas. Do you think that's a question on the next exam?
Yes.
Oh, guaranteed. Now, where did we say the catalase is stored in the cells? It's in the peroxisomes. Very good, good, good. So here are the experiments on the screen. The first one, we're going to test something called the substrate level concentration, the effect of substrate on enzyme activity. Can anyone tell me from that reaction that I wrote on the board, if you can capture that, Mason, on the board there, which one is the substrate? Nice, the hydrogen peroxide. This is equivalent to me saying which one is the reactant. And we talked about before, reactants are the things going into the reaction. What are the things coming out of the reaction? Products. Very good. Excellent. So hydrogen peroxide is the substrate of this reaction. Again, another guaranteed question on the exam, on the next exam. Because if you don't know that, then none of it will make sense to you. Okay, so we're going to look at the effect of increasing the amount of hydrogen peroxide on the reaction. Okay? The other thing that we're testing is temperature effect. What happens to the enzyme when it's put on 0 degrees, 20 degrees, 50, and 70? What will happen to the enzyme when it goes to 70 degrees? Yes, sir.
Will it break down faster or slower?
Break down what?
Faster. Substrate, right?
No. Listen to the question. What's happening to the enzyme at a hot 70 degrees?
It's moving faster.
What's happening to the enzyme? Do you want to sit in water at 70 degrees Celsius? I don't think so, right? Yeah, I don't think so. 70 degrees Celsius, I don't know about that. That's almost like boiling. You might handle it. You might like handle it with your skin, but your enzymes are like going crazy with that.
So you kill them, say?
Kill, meaning what?
Breaks it down.
Breaks it down. Watch very, very carefully. I'm so happy you said the word kill. Because if you kill an enzyme, you go to jail. There's no such thing as kill. What was the word that we used?
Diffusion.
Denature.
I heard it. Denature. You denature the enzyme. Because what's holding the enzyme? Which bond is holding the secondary structure of an enzyme? Got it. And how do you break a hydrogen bond?
Heat.
And that's what you're adding. Now we're catching it?
Yeah.
We talked about it. We said that in chapter 5, we said that the secondary structures of enzymes are held by hydrogen bonds. And we also said that hydrogen bonds can be easily broken with temperature. And that's exactly what you're doing. You're heating it up. Oh, that enzyme is not going to come near hydrogen peroxide because it's already denatured. Do not say dead. Do not say killed. All right? The scientific word for that is denatured. Its structure is misfolded. It doesn't work anymore because of chapter 1. When you destroy the structure, you destroy the enzyme's function. So you have to bring some information from other chapters to make sense of what's happening here. So that's what we're doing. We're showing you that with increasing temperature, the enzyme activity is going to decrease to almost 0, if not 0, because the enzyme is no longer working. Why? Because its structure is destroyed, which is called denature. Now, in the same chapter, we said that we can destroy or denature the enzyme in other ways. Another way is pH. A third way is salt, if you remember all that information. So we're going to do a pH test. We're going to see what happens to the enzyme activity when we change the pH. We'll see that. So those are the 3 tests there. Now, This one is a cool one. This one, we're going to test for the presence of the enzyme in blood versus yeast. Of course, we know now yeast is eukaryotic, it's not bacteria, and it is a fungus. Okay, we'll test. That's a simple test on the slide. It is so easy to do, and you'll see it. And when I go do it, maybe I'll do it right here so most of the people can see it. Make sure you can see it, because as soon as I add the substrate, which is what?
Catalase.
No,
Peroxide.
hydrogen peroxide. It's gonna bubble if the sample has the enzyme. What's the enzyme? Catalase.
Yeah.
So let's do that one first. This one right here. So you're gonna go to page number 63. You don't need to write observation, conclusions, and all that. You're gonna just make a statement. You're gonna simply say, the blood has or does not have catalase. Yeast has or does not have catalase. Well, we'll figure it out. We'll find out. So I'm gonna take a drop of blood. This is sheep's blood. It's simple. Just one drop. And they prepare all of this just for a single drop. I don't need all of this. Just need a drop. That's yeast. Good. Now, to confirm this, make sure you can see. Make sure you can see. So position yourself. All I need to do is add the substrate, hydrogen peroxide. I think that's the 4th time I said that. Okay? So look for the what? Bubbles. There it is. So do you see the blood bubbling?
Yeah.
Does that mean, what does that mean?
It has.
It has catalase. You gotta kind of look closely for the yeast. Come close here. Do you see it bubbling? Go look at it. While I go get something. You see the yeast bubbling? Yeah. So what's your conclusion statement? The blood has, the yeast has. We're done. So easy. Did you ever, when you were young, did you ever fall playing whatever, basketball, baseball, biking, and you scrape your knee, you run to mom, and she runs into the medicine cabinet, and she gets this, I think it would probably be like a brown bottle, hydrogen peroxide.
Yeah.
And then in the middle of your crying, you start laughing. Look at those bubbles, what is that? That is so cool. That's what it is. It's your blood that has the catalase. The bubbling was coming from your blood, because your blood has catalase too. But why do we use hydrogen peroxide? To kill bacteria. The concentration of the hydrogen peroxide that your mom put on your wound is so high. I mean, some bacteria can produce catalase to overcome the hydrogen peroxide, but the concentration that we use is so high that it kills all of them to keep the infection away. But because you're bleeding in the process, the catalase in your blood is the one that causes it to bubble. Okay? So that's a simple test. They both have catalase, and that's all you have to do is make that statement. Make sure you have that statement in there when you hand the lab in next week. All right. Now we're going to do the next one. The next one is the substrate, and that is going to be table number 1, page 64. Over here, you're going to write 3, 6, 9, 12, 15, 18 drops, just like you see here. Down this way. 3 in the first column. 3, 6, 9, 12, 15, 18 drops. In the second column, I'm going to give you the height of the bubbles. Which means, once I add the two together, it's going to bubble. What's the bubble? What gas is the bubble? Oxygen. And all I'm going to do is measure the height, how high the oxygen bubble column goes, which has a direct correlation with how active the enzyme is. Okay? Very simple. All right, so let's do this. Let's do the drops first. Drops of what again? Of hydrogen peroxide, followed by, okay, I'm going to do the 1 ml of catalase first. 1 ml of catalase in each one. So I'm going to add the 1 ml of catalase real quick to each one. Alright, here's the 1 ml of catalase. Now the first one is going to be 3 drops of hydrogen peroxide. Now, I want you to do me a favor. As soon as you see the bubbles, you're going to all say, wow. Okay, because that's as exciting as it's going to get. Alright? Promise me? Good. Let's see. Thank you. Only one okay, good. Let me see now. We're gonna record 13 millimeters. First one is 13 millimeters for the 3 drops. Good? What is your prediction? Will the column get higher or lower or stay the same?
Higher.
Yeah? Good. So 1, 2, 3, 4, 5, 6. Good. 25 millimeters. Wow. Okay. 25. Okay. 30. Hopefully you're writing these things down.
So why is it such a bigger change between 3 and 6?
We doubled up the drops.
Yeah.
We added more. We'll talk about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. The more substrate you add, the more the reaction occurs. So
69 wasn't as big as it changed. That's why.
yeah, it's not going to be like a linear thing. It's going to be more like this. You'll see. So we're talking about 35. Watch this. You'll see the graph right here. The graph that you're going to produce next week will be right here, right in front of you, visual, in real time. Watch this. The next one is what, 15?
Yes.
Yes.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We're talking about 39. The last one is 18. The next one is, I'm going to say 44. So tell me the number so I can write it down.
19, 25 for 6,
Uh-huh.
30 for 9,
Uh-huh.
35 for 12,
Uh-huh.
39 for 15, and 44 for 18.
Good. Okay, so look at the graph that you guys are going to produce next week, or during the week, to hand in next week. There it is. Do you see it? There it is. I mean, it's like in real time right there for you. Let me move it for the camera there. There's the graph right there. So the top of the bubbles there, that's gonna be like your line that you're gonna graph. You use line graphs. Next week, I want line graphs for everything, okay? So we'll leave that here. So what happened is, to answer your question, as I added more substrate, there's still enough enzyme to do more reaction. But if I did another tube, what's gonna eventually happen is, it's gonna go up, but then it's gonna level off. See, I wish we can do more too, to show you that effect. Sometimes I see it with these two. Obviously today I didn't see that. It goes up, and then no matter how much more substrate you add, you don't get any benefit. Why? Because all the enzyme is busy. All of them are busy. So you add more hydrogen peroxide. I can't help you with that. I'm overwhelmed. It's kind of like, sort of like what happens to us on a daily basis when you have 10 things to do every day. And someone says, can you also do 5 more things? Well, those 5 other things you will not do because you're overwhelmed doing the other 10 things, you see? Well, it's the same thing with the enzyme. It has its own like built-in capacity. I can only do so much. Don't push everything on me. So when we add more substrate, that's equivalent to I would add substrate, asking a very busy enzyme to do more. And the enzyme is going to say, no, I can't. I can only do this much. The other substrate that you add is going to stay as hydrogen peroxide. See, that answers your question. Okay, so that's the substrate concentration effect. Okay, now let's do this one, the temperature effect. Now, while you were taking the exam, while you guys were taking the exam, I set this one up. I put these two at 0 degrees. These two at 20 degrees, 50 degrees, 70 degrees. What are these two? Well, one of them has 1 ml of hydrogen peroxide. The other one has 1 drop of the enzyme. Obviously, I have to keep them separate from each other and get them acclimated to the temperature, get them used to the temperature. Now, that part is done. And all I have to do now is add the two together. Of course, it is much easier to add this one in this one. Do you agree? I'm going to drop this one in this one because it has more volume, of course. So, watch it happen. What is your prediction? 0 degrees Celsius. The enzyme is very cold. Will it happen?
Yeah.
It will not happen.
No.
Not this enzyme. Now, some enzymes can work at very cold temperatures, not our enzymes. That's why if you fall in a lake that is frozen, you better get out of it real soon because your enzymes will stop working for you, and of course, that's not good for you. So watch this. No bubbles, am I right? There's no wow. Okay, let's see. Yeah, nothing. We're gonna call that 0.
0 degrees.
0 degrees is 0. Yeah. All right, the next one is 20 degrees. Do you expect the bubble reaction to occur? Yep. I expect the bubbles there. All right, let's see. Oh yeah, lots of it. Look at that. Big, huge difference there. We're gonna call that one, that's a late ooh, okay, anyways. But I'll take it, 83. Look at that, 83. 83 millimeters. Everything is in millimeters today.
And we're doing Celsius.
Yes, these are Celsius. Okay, next one, 50 degrees. What is your prediction?
It'll bubble like crazy.
Well,
No, it'll bubble over.
it's on its way to denaturing. It might bubble. I don't think I'm gonna get this much. Let's see. I might be wrong. Let's see. Okay. It's gonna be 38. What was the 25?
Oh,
That,
20.
no, the 25 degrees, sorry, the 20 degrees was what? 80? 83. What was the 50?
38.
38, good.
And how do you get them to different temperatures?
Oh, we got water baths, that's good. We got water baths back there. Yeah. Okay, 50 degrees. Prediction.
Nothing.
Nothing. Why? Because the enzyme?
Denature.
Nice. Denature. Let's see. Oh, thank God. It did not embarrass me. I always hold my breath when I do this one. We're going to call that 0.
Let's 70 degrees?
That's 70 degrees. So when you do go graph it, look what it's gonna look like. Again, you can see it. You're gonna have a visual of it. And Mazen is gonna record it for everybody. Look at that. There's your graph right there. There's your graph. You notice something? It goes up and then it comes down. That top point, that's what we're going to talk about in the lecture. It's called the optimal temperature. The temperature, listen to the definition. Optimal temperature is the temperature at which the enzyme works the best. Very straightforward. This is obviously for our experiment, that's 20 degrees. Now, had we done 37, it might have been better. But we can't go there because we didn't do it. You can't assume anything. In science, you have to test it. I'm thinking this enzyme will work much better at 37, but I can't say that unless I test it. So for today's experiment, what is the optimal temperature of the catalase that you are using?
20.
It's 20. Again, the working definition of that, optimal temperature is the temperature at which you get maximum activity of the enzyme. What do you think the optimal temperature for your enzymes is?
97.
98.6 Fahrenheit. But what is it in Celsius? No.
32 something?
37. Your body is at 37 degrees Celsius all the time, I hope. Unless you're sick or whatever. All right, so 37 degrees Celsius. Most of your enzymes, optimal temperature is 37 degrees Celsius. Now, there are some enzymes in your body where the optimal temperatures is a little lower than 37, and that's for the men in the room. For men in the room, there are enzymes that are used in the production of sperm and hormones and things like that. They only work best at a temperature slightly less than 37. That's why the testicles are technically positioned outside of the body, in the scrotum. Because if they're inside the body, it's too hot. So one form of infertility, doctors sometimes, a man goes to a doctor and says, you know, I'm having trouble having babies and so forth, and the doctor literally says to them, go take a cold bath. And I mean a cold bath. Or simply wear loose clothing. Men that wear very tight pants and clothing, they tend to reduce the sperm count because the testicles are positioned in a position where the temperature is too hot. Even a slight difference in temperature can mean a lot. So they would literally tell them, wear loose clothing, or again, sit in a cold water ice bath kind of thing, but that's gonna be painful. I mean, not ice, but cold water, to drop the temperature and then get the sperm production going again. So this is actually a true fact. I mean, sometimes it happens, unless there's something more drastic going on there. This is just a side issue. That part is not on the exam. It's just a side issue, a really interesting part of why are the testicles in the scrotum technically outside the body? It fits. The correlation there is amazing. It's all about temperature here. The enzymes don't work at that high temperature for that purpose.
I have a question.
Yes, sir.
I heard you're not supposed to put your phone in your pocket for that reason.
That's another reason. Now the phone is because of heat, one thing, but the worst thing is that radiation. There are websites that tell you do not put your cell phone next to you at night when you're sleeping to charge, because you can have effects on your brain. But, you know, how do you receive a phone call? These are all waves, and it can cause problems in the long run. See, the ruling is out on that, because with time we're going to confirm if that is true or not. But do you want to be part of that story? No. No. All right. Okay. Let's go on to the next one. The next one is the pH. So I need the empty tubes for that. Yeah. Okay. We're doing a pH test now. We have hydrogen peroxide in different pHs. So we're not going to use this hydrogen peroxide. We're going to use the hydrogen peroxide in the corresponding pH. So what it asked us to add 1 ml of each one of those and then follow it with a drop of catalase. So I'm going to do that first. pH 2. So now we're not looking for the optimal temperature anymore. What do you think we're looking for? The optimal pH. Yeah.
The first one was pH 2.
The first one is pH 2. The next one is pH 4. The third one is 7. I think you see it on the screen, right? 9. And what's the next one? 11. 11 is trouble. Oops. I'm just trying to stick that in there. Okay.
Can I give a prediction?
Huh?
Can I give a prediction? I think like the more acidic it is, the less enzymes in the word.
Yes, you're absolutely right. For certain enzymes, the more acidic this is, it will die. Not die. Sorry, I made a sin. It will denatured. All right, it will denature. Okay, but for some enzymes, in other enzymes, the more basic, the more it will die. Again, denature. I know it's the default thing to say is die and kill. I understand, but it's the denature. But there are some enzymes that work best at pH 7, pH 6, or whatever. Enzymes are different than temperature. All of your enzymes work at 37 degrees Celsius because that's what you are. But in certain parts of your body, your stomach is a pH 2.0. Those enzymes work best at 2.0. You put them at 8.0, they don't work. Right outside of your stomach, in your intestines, the pH is a little bit more basic. So it will work at that pH, you see? Now it depends on where in your body is the enzyme. And of course it depends on which enzyme. This is catalase. Let's see where the optimal pH is gonna be. I'm gonna add 1 drop of catalase. Okay, pH 2. 1 drop of I gotta get rid of the air. Ooh, nothing. We're gonna call that 0. That was not impressive at all. So we're gonna call that 0. And I make sure I don't, okay, get rid of the air. Didn't want to let go. All right, here we go. So that one, we're gonna talk, we're gonna say 27. 27. Okay. This is the pH of what? 7, right? Whoa. All right. We're talking about 75. pH 9, a little bit. So it's starting to denature. 35. This is trouble. This one is trouble. We're not supposed to have any, technically, but we might still get some. Yeah, we still get some. I'll tell you something. It's stayed at, it's actually 34. We're pretty close. We should go actually to pH 12 or 13 to really denature it. It's still kind of active between 9, 10, and 11. We need to go to at least 12 or 13 to really denature it. But again, here's your graph. It is bell-shaped curve. It goes up and it comes back down. So what is the optimal pH? 7.0. Again, the definition of optimal pH is the pH at which you get maximum activity of the enzyme. That's what it is. I'll say it again. Optimal pH is the pH at which you get maximum enzyme activity. So, your job now for next week, you have to hand this in next week, is to graph all the data that you wrote down. Graph all of them. Watch out, please. Make sure you label the axes. Make sure you know which one is the dependent variable and which one is the independent variable. This one will always be the y, and this one will always be the x. You'll have the video to follow up in case you forget what I'm saying here. So let's quickly run through this. Number of drops versus enzyme activity. Which one will go on the y-axis? Got it, enzyme activity. So your results will go on the y-axis. That means the number of drops will go on the x-axis. You have to label it. You have to equally space it. You have to use the whole thing. Now, I have some students who draw everything in this little, right here. I have to put it under the microscope to see it, all right? And I don't have a microscope at home. So I'm gonna mark you wrong. Then you're gonna make a statement right here about the results. Okay, let's flip the page. The y is the dependent variable. Yeah.
Would you say the definition of optimal pH is again?
Optimal pH is the pH at which you get maximum enzyme activity. It's exactly like the optimal temperature. You're just taking the word temperature and replacing it with pH. Okay. In this one, where do you think temperature is gonna go? The x-axis. And the enzyme activity will go on the y-axis. You label both axes. You make sure you equally space everything. Make sure you use the whole graph. And you're neat with your graphing. I will mark you down if it's not neat. Again, you make a statement about your data on the bottom. pH. Where will the pH go? X-axis. Enzyme activity on the y-axis. Again, use the whole graph. Now, what's the maximum number are you gonna have for pH?
14.
You got it. You have no clue or idea of how many students go 0 to 50 on the pH. There is no such thing. The maximum number is 14. So you can go ahead and scale it 0 to 14 if you want. But the maximum number that we use today is 11. You don't even need to go to 14. So you can go, for example, 0 to 12. Okay, everything good? Do when?
1 week.
1 week from today. Thank you, mate.